In this study, the investigators set out to attain a better understanding of the role that TLR3 plays in the pathogenicity of influenza A virus, as well as its role in the host immune response during infection. One of the most severe complications of influenza is acute pneumonia, which is a serious respiratory disease consisting of inflammation of the bronchoalveolar spaces in the lung tissue. The disease is characterized by fast progression as well as having a high mortality rate. Therefore, the researchers were interested in understanding in more detail the molecular mechanisms associated with the pathogenesis of influenza virus, as well as the related immune response it triggers in the host, in order to develop better prevention and treatment for influenza. Before diving into the details of this study and their findings, I would like to refresh our memories about exactly what toll-like receptors are. Recall that the innate immune system is the first line of defense against infection. In order to recognize materials as foreign, cells are equipped with special receptors found both at the plasma and endosomal membranes. Their purpose is to bind non-cellular molecular patterns such as double-stranded RNA. TLRs are members of this large group of pattern recognition molecules, and many different types of TLRs have been identified in various cell types. Their mechanism of action is once a TLR binds a pathogen-associated molecular pattern, such as double-stranded RNA by TLR3, a signal transduction begins that usually results in changes in gene expression. For example, secretion of interferon or secretion of other cytokines that initiate an inflammatory response. This in turn results in activation of the adaptive immune response. Now that we have reviewed what TLRs are and their function, it would be a sensible assumption that TLR3 would act as a protective component during an immune response. Indeed, the investigators in this study, at the beginning of their research, expected to collect data that would support such assumption. After challenge with influenza A virus in wild-type mice and TLR3 deficient mice, they determined several dynamic parameters of the disease, including survival rate, leukocyte recruitment to airways, respiratory distress, secretion of inflammatory mediators, and viral clearance. However, to their surprise, they found that the mutant mice lacking TLR3 had an unexpected advantage over the wild-type mice. The researchers first wanted to determine the time course of the major dynamic parameters of influenza virus-mediated acute pneumonia in wild-type infected mice. Thus, each mouse was inoculated intranasally with a lethal dosage of 300 PFUs of the virus. Figure 1A shows that all wild-type mice died within 12 days. Interestingly, when they later inoculated the TLR3 deficient mice, they found that they exhibited an increase in survival of 26% after challenge with the same dosage as the one administered on wild-type mice. We can see in figure 2B that TLR3 deficient mice died 3 to 5 days later than controls. Now, they also perform additional survival tests with lower virus doses of 100 PFUs and 30 PFUs. In this case, they observed that clearly the absence of the TLR3 receptor protected the mice from death, as their survival increased with the decrease of viral challenge at 100 PFUs. But at 30 PFUs, none of the mice, neither wild-type nor TLR3 deficient mice, died. Next, they wanted to determine the role of TLR3 in respiratory dysfunction, as well as the total amount of protein and secreted inflammatory cytokines found in the lungs. To do this, they analyzed bronchoalveolar lavage fluids, where samples were taken from both groups at day 3 post-infection, since in both cases the viral load appeared to peak at that time. Figure 3 presents measurements taken before day 0 and day 3 after infection. Figure 3a shows the PEN index. This is a measurement of respiratory distress derived from the respiratory capacity of each mouse, calculated mathematically from the expiration and relaxation times. Figures 3b through 3d shows the total protein content found and the amount of activated T cells and the levels of interleukin-6 in the sample lavage fluids. For clarification, interleukin-6 acts mainly as a pro-inflammatory agent, and it has been usually associated with influenza virus infection in pulmonary inflammatory diseases such as pneumonia. We can definitely appreciate that for all the measurements taken, TLR3 deficient mice compared to controls had significantly lower values at day 3 post-infection. Subsequently, the bronchoalveolar lavage fluids were further analyzed at day 9 post-infection to look for the presence of various inflammatory proteins, to get a sense of whether TLR3 was directly regulating additional mediators. Therefore, they focused on samples from the TLR3 deficient group. In Figure 4a, we can see that there is a clear reduction of the expected inflammatory mediators such as interleukin-6 and interleukin-12p40. Interestingly, expression of other components was increased, and of particular interest to the investigators was the higher levels observed of interferon gamma. So they subsequently confirmed this finding via an ELISA, where it showed that TLR3 mutants had 14.5 times higher amounts of interferon gamma than the wild-type mice. To get a visual appreciation of the physical damage caused by influenza A virus-induced pneumonia, lungs were harvested from infected and non-infected wild-type and TLR3 deficient mice. We can see in Figure 4c that the lungs of infected wild-type mice were severely injured with evidence of hemorrhage based on their dark surface. Contrary was the case of infected TLR3 mutants, where their lungs seemed to only present faintly diffuse red lesions. These observations suggest that the absence of TLR3 resulted in less injury by influenza A virus-induced pneumonia. So far, the results have shown that a strong inflammatory response takes place in the lungs of wild-type mice after influenza infection, while a significantly reduced or different reaction takes place in the TLR3 deficient group. A commonly accepted view is that inflammatory responses can act as a defense mechanism in the host. But as the authors mention in their discussion, in some cases it has been seen that viruses can exploit these pathways to enhance their own replication. Therefore, using a quantitative PCR technique, the investigators were concerned next with determining whether the TLR3 dependent host response could play a role in influenza virus replication. We can see in Figure 5 that viral replication was efficient in both experimental groups, having similar titers from day 1 to day 4 post-challenge. However, at day 9, the viral load was significantly reduced in wild types, while in TLR3 deficient mice, the virus remained significantly higher, approximately 9 times more than the controls. These findings suggest that the attenuated secretion of cytokines correlates with decrease in disease symptoms, but not with a lower viral load, since virus replication remained high. Finally, the authors set out to find out which cell type participates in the TLR3-mediated detrimental immune response. In other words, the upregulation of TLR3 during infection directly results in upregulation of which immune cell type. They collected leukocytes from bronchoalveolar lavage fluids from both experimental groups at day 9 post-infection, and cells were characterized through flow cytometry. For reference purposes, leukocytes from naive mice were analyzed, which contained approximately 90% of macrophages. Lavage fluids from wild-type infected mice contained mainly T lymphocytes, where the most abundantly found were CD8 cells, followed by neutrophils, and in lesser amount, macrophages. This was completely different than the results obtained from infected TLR3 deficient mice, where the population of CD8 cells was reduced by 1/3 compared to wild types. Also, the number of macrophages was about 1.5 times lower, but the amount of neutrophils found was at least 1.5 times higher in TLR3 mutants than in wild-type mice. CD4 cells showed no significant difference between the two infected groups. Now, we learn in this course that interferon is a key player in the host immune response, where it is capable of activating several different pathways. However, the authors stated in their discussion that recent studies have shown that specifically interferon gamma, which was significantly higher in TLR3 mutants as we saw in figure 4a, may have negative effects on CD8 cells by either inhibiting their proliferation or possibly inducing their apoptosis. And we see clearly a connection here between higher levels of interferon gamma and lower CD8 cells in TLR3 deficient mice.